0?
So it's actually just normalized, right? So it's just interface minus the center.
Oh.
That's all, right? So that you're at 0. Okay, this is the chlorophyll. It doesn't change that much. All right, and then, all right, so this is where we are with this part of it, right? So we think that these hypoxic zones are dependent on microbial activity because we can prevent them with antibiotics or by algal removal, right? It doesn't mean that the idea of the allele chemical studies that Rasher and Hay are pushing, right, where you get things coming in that actually directly kill the corals, they can also be happening. Does that make sense, right? And they would complement, they would reinforce each other. So remember that. This is the mechanism as we see it, and I think that that's only that's really important in that initial first step when we're moving towards turf algae. They have unique microbial communities, and the animal is stressed along those things. Okay? All right. So you guys should. This is the new. This stuff is summarized in this basic this review that just came out, and really what we're talking about here is a lot of what I just showed you. Plus, we're starting to bring into play actually how these dynamics are going to look. And it's worth, going in here and looking at this because this tells you about the different boundary layers that are going to be associated with these systems and what we expect to be happening in them. So this is actually worth, remember any system like this, you're going to actually have a place where everything is by diffusion, right? So there's going to be some point where everything is working by diffusion. And so that makes it more complicated to think about, and why probably you have to do some of the studies in more controlled things. You can't just be doing things out on the reef. You'll never figure it out, I'll bet you. All right, the other things I just should give you a feel of what we're looking at. Okay, so this is these studies from that Allison and Andy. So remember, Allison's making these optodes, and these detect oxygen. And what you can actually see is in these experiments is that this is an example when it's photosynthesizing, the algae is photosynthesizing. You can actually see where the bubbles of oxygen are running up through the system. Okay? And this is what it looks like when you turn on the current, right? So then, not surprisingly, there's all this oxygen running down. Presumably, that's where all the DOC is too, right? So this is like the world's best spot to be a microbe in many ways, right? Because you'd have a lot of oxygen and you'd have a lot of DOC, okay? And and this is the hypoxic zone associated with it at night, okay? And then finally, there is evidence of this kind of on a reef, and so this is a different study, but you guys need to really be aware of it. So this is Kristen Marhaber's stuff, and basically what she was doing is she was putting coral larvae out and looking at survival on a reef, and she was doing an up current and down current of coral head, and this is smart. Okay, so think about this. It's specific to this is the Jansen-Connell hypothesis, which basically says that if you want to be away from your parent because your parent is more likely to have your parasite with you. Okay? So she did this on the same species or different species. So she has the larva from the same species down current, or she has the larva downstream of another species of coral. Okay? And it is specific. So being a little larva next to next to your parent is a bad idea, effectively. Okay. And what's interesting for us is that this is basically the probability of the larva surviving. And you can see it goes down the closer you get to the coral, except for this weird spike right here, which nobody quite understands. Okay? It could be that there's an eddy or something going on right here that basically makes this a better place. So a larva can actually be really close upstream of the coral, at least in this case. And then as you go downstream, it gets worse and worse, and then it tapers off, right? There's something going on there. And the microbial activity, so this is just total number of microbes. It kind of correlates, but it's not really perfect. Okay, so these are just the cell microbes per cell in the water, or microbes per mil in the water column. And this drops off in this area, and then it's higher, of course, when DOC is coming off, which would match stuff. And then there's like this increase out here. And that could be that maybe there's an eddy or something coming off, but see, it's really hard to understand this right now. So nobody has these visuals down. And it's not clear that the fluid dynamics explains this, but it does look like you can actually see these long range, these are actually pretty long range effects of having a coral in a particular place.
What is that scale?
This is actually quite large. That's probably centimeters.
Okay.
I'll bet you it's centimeters, yeah. So that would be 2 meters. Yeah, okay.
So
Yes.
has anyone done with Alice and stuff with the, all the DOC washing down, has anyone done like current-based sort of survival? Like if you have turf upstream of you, because there are prevailing currents on the reefs, right?
Right, no, not yet. So that's exactly what
That'd
we
be
have
really
to
cool.
figure out.
Like at a
right?
nice little entry into a lagoon or something?
Yeah, exactly.
Yeah.
So you need that, exactly. We really need to get this figured out, because you can see that obviously we should be able to make the prediction, but it's actually understanding the swirling of water in a crop that's really difficult at this point. And then basically where we're headed to get to some of this stuff is basically we're going to, I think what we're going to end up is with a map between, and by we, I mean, Stewart's lab and Zawada and these other people are interested. So they're getting big tile arrays of things. So they're taking big swaths of reef, and so like Zawada gets LiDAR things, so he gets like 100 kilometers of reef, and he's got like, in those cases, down to, let's say, decameter sort of level pictures or information. And then they're, then if you drag these cameras across it, then you can get down to maybe centimeter level resolution of these interaction zones. And then of course, we've got all our stuff that will feed into this, where you can imagine, so these are the hyperspectral ones, which are really pretty cool, because you can see where coral and algae are fighting it out. You can see the cyanobacteria living right there. There's all this cool stuff there. Then we've got these microbiological views, which is, Eric's stuff is coming up, as well as the transcriptomes will be coming into this. And then, the EMs. The EMs, I think, help a ton, right? This one I just love. That's the scrubby bubbles on the coral. But, you like what I showed you with Linda's stuff, for example. And then finally, there's the normal views. And I think that these things are getting, I think these surveys, like we did with Katie's stuff, is actually really important out there, and so we should keep doing more of those. And these visual things, like what Laura's doing, looking at what actually lives at the interaction zones, is at kind of the macroorganism. And right now, the thing to tell you is hermit crabs are important, right? We don't know why, and we don't understand anything about them. But these guys are the main players in the Line Islands, at least. Okay, at these interaction zones. Okay. And then, and they're all happening at night. Right, this is all a nighttime thing. So it's very cool. I have no idea what's going on. And we see no evidence either of predation on the hermit crabs, which is another thing. So like what's going on hermit crab wise. And also, I don't know if you guys remember, but the hydrophone work showed that there's that's where all that noise on the reef is coming from at night. It's basically the hermit crabs walking out, and it's probably the shells heating up against the coral.
Yeah, snapping.
It's not the snapping. No, that's a completely. Yeah. So everybody says snapping shrimp. It has nothing to do with snapping shrimp. It's actually all due to hermit crabs, and probably either their little claws walking along, or their shells hitting up against the coral is probably what's going on. And that's interesting from the hydrodynamic, the hydrophone people, because they actually didn't know any of that up to this point. Okay. All right, so can you guys, let's just keep going, because this one's almost done, and then it'll be done as a thing. I just want you to remember, if we go back to this view of the reef, there is I told you nutrient additions did kill off the reef, okay? Or kill off the coral holobiont. That doesn't mean they aren't bad for a reef. And you can imagine because they actually could feed the algae here. Okay? And that's basically, I think, exactly what Linda saw in the Line Islands data. So again, here we are. So it's important to remember here that we're in the southern Line Islands where we first started this work, okay? Which are there are tons of grazers on these systems, right? These are like some of the highest biomasses of sharks and grazers ever measured, like fish biomass for a coral reef. Okay? So this is the best thing. So you have as much grazing as you can possibly get done. Okay? The other important things from the geochemical point of view is this is the part of the ocean where there's essentially no iron coming in because iron has to come from land and has to be blown in. So this is this high nutrients, low chlorophyll regions of it, and the limiting nutrient is iron. And these are carbonate platforms, so there's absolutely no land sticking up. So there is no iron out here. And this is where we get these block reefs, and these block reefs are where there was a shipwreck. Okay? And they're